Most of the data that we have in advanced stage Hodgkin lymphoma derives from clinical trials and a few retrospective studies, particularly from European and North American academic centres. So there's really a paucity of data from real-world evidence. And we know that real-world data is important because it hopefully recapitulates the data that we've seen from clinical trials, but also because regulatory agencies and reimbursement authorities are now looking for real-world data in order to better inform their decisions around drug approval and reimbursement. So this study was designed to look at advanced stage Hodgkin lymphoma, particularly relapsed refractory Hodgkin lymphoma, but also first-line therapy, and data was collected over a 4-year period across 13 countries in 6 major jurisdictions and regional areas. So it was highly representative because 1,600 patients with classical Hodgkin lymphoma were identified and more than 400 patients with relapsed Hodgkin lymphoma were identified. And so I feel that it was truly reflective of real-world data and clinical practice. So I think the data was collected was very informative. And when we look at relapsed refractory disease, it illustrated a number of key points. So when we look at relapsed refractory Hodgkin lymphoma, only about 50% of patients survive after 3 years, despite high-dose chemotherapy and stem cell transplantation. And having learned that from clinical trials, this was recapitulated clearly in the evidence that was obtained in the Be Holistic study. So in relapsed refractory Hodgkin lymphoma, we've learned some very important data that in many ways is reflective of clinical trials, and that was reassuring, particularly in both relapsed refractory and of course first-line disease. If we want to stick to relapsed refractory disease, it's quite sobering. Patients, about nearly 90% of patients were able to receive intensive salvage chemotherapy. But of those, only 62% achieved a complete or partial response, and indeed only 32% achieved a complete response. Furthermore, of the patients who were deemed eligible for high-dose chemotherapy and stem cell transplantation, and that was about 70% of patients, only 3/4 of those ended up getting to transplant. So overall, only 50% of patients got to transplant. Now, there are a number of key factors that were prognostic in determining the outcome of patients. That outcome was sobering because at 3 years, progression-free survival was only 40%. And that's pretty much reflective from clinical trials in the literature, perhaps slightly inferior, but it's around the 40 to 50% mark. Clearly, there is an unmet need. So what we've learned in the relapsed refractory cohort is that a number of adverse prognostic factors were identified. For example, the Josting score was confirmed as being prognostic since it included 3 factors. Those patients who relapsed early, that is within 3 to 12 months, those patients who relapsed with advanced stage disease, and those patients who relapsed with anemia. But the factor with the highest hazard ratio was in fact those patients who achieved an inadequate response to their salvage therapy. And that hazard ratio was high at just under 6. And it's here where we clearly have the unmet need. That response rate and that adverse risk factor has been borne out in recent studies with the advent of PET imaging after salvage and before transplant. And so it's concordant with those results, which have told us that those patients who have a complete metabolic response to salvage therapy and then go on to transplant do very well, with progression-free survival at 3 to 4 years of around 75 to 80%, whereas those patients who do not achieve a complete metabolic response to their salvage have a poor outcome after transplant with progression-free survival of only 25 to 30%. So it's that group that represents the clearest unmet need. That is, those patients who have an inadequate response to salvage therapy prior to going on to transplant. We need to improve that response rate, improve that complete metabolic response rate so that more patients can go on to transplant. Now, to date, we haven't had very good agents in order to do that. To date, we've only had really 3 modalities. That is, another cross-resistant chemotherapy regimen. We've had radiation therapy. And in some jurisdictions, we've had tandem transplant. But now, with the advent of novel agents, it's clear that this is where their role will become most important. And it has been demonstrated already with the use of novel agents such as PD-1 inhibitors, with toxin-coupled antibodies such as brentuximab vedotin, that using those agents, either alone or in combination with chemotherapy, either single agent such as bendamustine or combination chemotherapy such as DHAP or ICE, that more patients can be rescued to achieve a complete metabolic response and then go on to transplantation. The impact is that it's first recapitulating the data that we've learned from clinical trials, particularly around, as I said, PET-assessed responses to first-line salvage therapy. And it's shown in the real world that we have similar inadequate rates of complete response to salvage therapy, here only 32%, and only 50% patients going on to transplant. So now we have the opportunity to use novel agents, as I said, alone or in combination, to increase the number of patients achieving a complete response, this time to a second-line salvage, or possibly even a first-line salvage, so that more patients can go on to transplant in remission, in a complete metabolic response, and therefore have a greater likelihood of long-term disease-free survival and cure. I think the other thing we learned from this study was about first-line therapy, because we collected data on 1,600 patients with advanced stage Hodgkin lymphoma being treated with first-line therapy. What it showed is across all these jurisdictions is that the regimen ABVD represented the standard induction therapy in well over 85% of patients. Again, consistent with treatment across Northern America and Europe. Not only that, but the results in the use of ABVD chemotherapy were very similar to those results seen in recent clinical trials, such as the Echelon-1 study, such as some of the PET-directed studying, like RATHL and the SWOG study, in which patients commenced on ABVD. So I think that's reassuring that patients being treated in these jurisdictions in countries around the world are achieving outcomes very similar, if not identical, to those that were seen in some of the big trials conducted in advanced stage Hodgkin lymphoma in, as I said, in Europe and North America. In recent years, we've really had to rely upon either phase 3 randomised studies or phase 2 studies that have been conducted in large academic institutions, particularly in Europe, North America, and here in Australia. And that doesn't always reflect the real world patients that we see in the clinic, because they have specific inclusion and exclusion criteria, and because sometimes you might have to wait in order to have the patients enrolled. Meanwhile, they might be progressing and declaring themselves ineligible. And I think it's the real world data that is extremely important in providing information about our patients in the clinic, not only to benefit clinicians, but as I said, to benefit regulatory authorities, because nowadays they are wanting more and more real world evidence when it comes to drug approval and registration, and when it comes to reimbursement by the authorities in different jurisdictions. So I think it's studies like this that are going to become more and more important, and we are already learning that, particularly in jurisdictions like Australia, where cost effectiveness is important, and it's the real world data that can often provide that cost effectiveness when submissions are being made for drug approval. I think this was the most important study of real world data in Hodgkin lymphoma presented at ASH. This is 1,600 patients with first line and 430 patients with relapsed refractory disease. So there's nothing else that recapitulates that. There are other important studies now looking at trying to work on the back of the results of these studies in order, as I said, to improve the results that we've identified in these studies in terms of salvaging more patients successfully, getting them into remission before transplant, and getting more patients to transplant. There are a few studies recently of, for example, brentuximab vedotin combined with chemotherapy agents such as bendamustine, or combination therapy regimens such as ICE, DHAP and ESHAP, that have been shown to be very effective in improving the complete response rates and therefore improving long-term progression-free and overall survival for those patients being salvaged and going on to stem cell transplantation. So I think it's going to be now the combination of these agents, both the checkpoint inhibitors such as pembrolizumab and nivolumab combined, either with another oval agent or with chemotherapy, and then the toxin-coupled antibodies such as brentuximab vedotin, as I said, particularly combined with either single agent or combination chemotherapy regimens. This study was a huge undertaking, as I said, because it involved 13 countries, 6 regions, including Latin America, the Middle East, South Africa, East Asia, Australia and Russia. And I think this was a huge undertaking to collect this sort of data. I think it's going to be very informative for future trials to have this data, and for regulatory authorities as well. I think the only potential disadvantage of the study was that we weren't able to collect in a comprehensive manner adverse event data, but that's not surprising when you're doing a study like this, but it certainly doesn't detract from the key messages of the study around patient outcomes in the real world. So I think investigators need to be commended for the comprehensive nature of the patient baseline characteristics, the patient outcomes, the details around their chemotherapy regimens, their response rates and their survival. And I think really it's to be commended, because it will be very useful in the future when conducting and designing clinical trials and when submitting to authorities for regulatory approval of new agents for both registration and reimbursement.